Welcome to our deep dive into anthrax. Now, I know as a medical microbiologist, you're already familiar with the basics, but today we're going to try to unearth some of the more intriguing details about this deceptively simple bacterium. We've got MicrobeWiki, some CDC reports, and even a fascinating case study from Microbe Canvas to kind of guide us.
You know, it's interesting how anthrax often gets portrayed as this highly contagious threat, but the reality is it primarily affects animals. Human cases are often tied to very specific circumstances and exposures.
Exactly, and that's what we're going to explore. So let's start with a quick overview from MicrobeWiki. We're talking about Bacillus anthracis, a gram-positive rod-shaped bacteria that forms these incredibly resilient spores. These spores can just hang out in the soil for decades, just waiting for the right opportunity to reactivate.
And that resilience is really all thanks to the spore's unique structure. It has a tough outer coat made of keratin-like proteins, a dehydrated core, and even specialized DNA-binding proteins to protect its genetic material. So this makes them resistant to heat drying and even many disinfectants.
Wow, so how did these hardy spores actually cause disease?
Well, animals primarily grazing herbivores ingest or inhale the spores while they're foraging. This is where those seemingly harmless spores transform into actively replicating bacteria, leading to a systemic infection.
So then humans were usually not the intended target, right?
Correct. Most human cases are linked to contact with infected animals or contaminated animal products. The CDC reports highlight agricultural regions as hotspots for anthrax, places where livestock farming is common.
Okay, that makes sense. Now let's delve into the different ways anthrax can actually manifest in humans. The most common form, cutaneous anthrax, has that classic black Escher lesion. I've always found that so fascinating.
It is quite distinctive, and it's worth noting that unlike a lot of skin infections, the Escher itself is painless. This can sometimes lead to a delay in diagnosis, as patients may not seek immediate medical attention.
Right, and that delay can be dangerous because even though cutaneous anthrax is typically localized, those potent toxins can still cause systemic complications.
Absolutely. Bacillus anthracis produces three main toxins: protective antigen, edema factor, and lethal factor. As the name suggests, the lethal factor is particularly worrisome. It disrupts cellular signaling pathways, leading to cell death and ultimately organ failure.
So let's move on to respiratory anthrax, which thankfully is much rarer. This form presents a unique diagnostic challenge because the initial symptoms mimic a common cold or flu.
Yes, that first stage can be quite deceptive. Patients might experience fever, cough, and just general malaise, making it easy to misdiagnose as a routine respiratory illness. But what's crucial to understand is that as the infection progresses, it shifts dramatically.
And that's when things become really serious.
Unfortunately, yes. The second stage of respiratory anthrax involves severe respiratory distress cyanosis and even hemorrhagic mediastinitis. The CDC reports highlight a stark reality. The mortality rate for respiratory anthrax is alarmingly high, even with aggressive treatment.
Wow, this really underscores the importance of early diagnosis and intervention. Now, another form, gastrointestinal anthrax, is even rarer, but often presents with particularly gruesome symptoms.
It does. This form typically arises from consuming contaminated meat. While thankfully rare, the clinical picture is quite severe. Bloody diarrhea, intense abdominal pain, vomiting blood. It carries a high mortality rate, emphasizing the need for prompt recognition and treatment.
Speaking of unusual presentations, injection anthrax poses a new challenge for us in the medical field.
It does. It really highlights the evolving nature of infectious diseases. This form is primarily associated with injecting contaminated drugs. What makes it particularly challenging is that the clinical presentation can mimic other soft tissue infections, leading to misdiagnosis and delays in appropriate therapy.
So unlike cutaneous anthrax, where that black Escher provides a visual clue, injection anthrax can be harder to pinpoint.
Precisely. And the CDC reports emphasize the importance of a thorough patient history, including any history of injection drug use to guide diagnosis and ensure appropriate treatment.
Now let's shift gears a little bit and talk about a specific case study that really caught my attention. It's from Microbe Canvas, and it involves a rather unexpected source of anthrax infection: hippos.
It's a fascinating case that demonstrates the complex interplay between environmental factors, animal health, and human disease. This particular outbreak occurred in Zambia back in 2011 during a severe drought.
And how did the hippos become involved in this outbreak?
Well, the drought forced the hippos to graze in areas they wouldn't normally inhabit, seeking out any available vegetation. Unfortunately, these areas happened to be contaminated with anthrax spores. The hippos ingested, the spores became infected, and subsequently died.
And then humans entered the picture.
Sadly, yes. Facing food shortages, local communities saw these hippo carcasses as a potential source of sustenance. This led to people consuming the contaminated meat and subsequently contracting anthrax.
It's a tragic example of how environmental factors can contribute to these kinds of outbreaks, especially in areas where anthrax is endemic.
And it raises a lot of questions for us as medical microbiologists. How do we balance public health concerns with the realities of food insecurity in these regions? How can we improve surveillance and early warning systems?
These are all important questions, ones that we'll revisit later in our deep dive. But for now, let's turn our attention to the topic of vaccination.
That's a good segue. The anthrax vaccine plays a crucial role, particularly for high-risk individuals.
It's fascinating, and I'd love to dig a little deeper into its development and efficacy.
Well, the current vaccine is based on a purified protein component, specifically the protective antigen from Bacillus anthracis.
So unlike some other vaccines, it doesn't contain live attenuated bacteria,
Right.
or even whole inactivated spores, right?
Exactly. This makes it a safer option for most individuals. However, it does require a multi-dose series for full protection.
And what about its efficacy against the different forms of anthrax that we've discussed?
The CDC data clearly indicates that the vaccine is highly effective against both cutaneous and inhalational anthrax. This is crucial considering the severity of these forms.
But you mentioned earlier that injection anthrax is a relatively new challenge. Do we have any data on the vaccine's effectiveness against that specific form?
Unfortunately, the data is still limited. Due to the relatively low number of injection anthrax cases, it's definitely an area that warrants further research and investigation.
Now, shifting back to the hippo case study, it raises some important questions about public health interventions in regions where anthrax is endemic.
The situation in Zambia really highlights the need for a multifaceted approach. That goes beyond vaccination. While vaccinating livestock can help reduce the risk of animal-to-human transmission, it doesn't address the underlying issues of food insecurity and limited access to healthcare.
So what other strategies might be effective in these situations?
Well, improved surveillance of animal populations is critical. By identifying areas and seasons where anthrax outbreaks are more likely to occur, authorities can implement targeted interventions.
That makes sense. Early warning systems could help notify livestock owners and communities about potential risks.
It's exactly coupled with educational campaigns about the dangers of handling animal carcasses, especially during those high-risk periods.
And I imagine that providing clear guidelines on safe carcass disposal would also be crucial to preventing further contamination of the environment.
Absolutely.
Okay.
Every step taken to break the chain of transmission can make a difference.
But realistically, in resource-limited settings, achieving these goals can be a challenge.
It's true, but even small steps like providing basic training to community healthcare workers on recognizing the signs of anthrax can have a significant impact.
Now, while we focus primarily on the natural occurrence of anthrax, it's important to acknowledge its potential as a bioweapon.
The deliberate release of anthrax spores is a serious concern, and the events of 2001 in the United States serve as a stark reminder of its devastating consequences.
Thankfully, we now have more robust protocols in place for rapid detection and response in the event of such an attack.
That's right. Advancements in diagnostic techniques like PCR-based assays allow for quicker identification of Bacillus anthracis. This speed is essential for initiating appropriate public health measures, including treatment with antibiotics and potentially post-exposure prophylaxis with the vaccine.
And speaking of the vaccine, its role in biodefense preparedness is undeniable. Ensuring adequate stockpiles and having clear distribution plans in place are critical components of any bioterrorism response strategy.
Absolutely. And beyond stockpiling, ongoing research into new anthrax vaccines and therapies remains crucial.
This includes exploring novel vaccine delivery methods, like oral or intranasal formulations, which could potentially simplify mass vaccination campaigns.
And research into new antibiotics or therapeutic antibodies could be vital in combating antibiotic-resistant strains of Bacillus anthracis should they emerge.
So as medical microbiologists, we're really at the forefront of this battle against anthrax. Whether it's in the context of natural outbreaks or the threat of bioterrorism,
Our expertise in identifying, characterizing, and understanding this pathogen is more critical than ever. And this deep dive, I think, highlights the importance of staying informed about the latest developments in anthrax research, diagnostics, and treatment.
Now, before we wrap up, I wanted to circle back to something you mentioned earlier, the challenges of balancing public health interventions with the realities of food insecurity in regions where anthrax is endemic.
It's a complex issue with no easy solutions. But as medical microbiologists, we have a responsibility to consider the broader context in which these outbreaks occur.
It's a difficult situation. On the one hand, we have a responsibility to protect public health and prevent the spread of anthrax. But on the other, we can't ignore the very real needs of communities facing food insecurity.
Absolutely. Simply telling people not to consume meat from potentially infected animals, that isn't always a practical solution. We need to think about sustainable and culturally sensitive interventions.
So what might those interventions look like?
Well, for starters, we can work to empower communities with knowledge. Educational campaigns can teach people how to recognize signs of anthrax in animals and how to handle carcasses safely.
That's a good starting point. But as we've discussed, education alone may not be enough.
Exactly. In some cases, providing alternative food sources during outbreaks could be crucial. This might involve distributing food aid or supporting local agricultural initiatives.
And what about the role of vaccination? Could vaccinating livestock play a part in reducing the risk to human populations?
Absolutely. Widespread livestock vaccination can significantly reduce the incidence of anthrax in animals, which in turn lowers the risk of human exposure.
It's about breaking that chain of transmission.
Precisely. And this highlights the importance of collaboration between different sectors: healthcare, veterinary medicine, agriculture, and even social services. A multidisciplinary approach is essential for tackling this complex issue.
It sounds like we need to move beyond thinking solely in terms of treatment and start thinking more about prevention and long-term solutions.
I completely agree. Anthrax isn't just a medical problem, it's a socioeconomic problem, an environmental problem. We need to address all those facets to truly make a difference.
Well, this deep dive has been incredibly insightful. We've covered a lot of ground, from the microbiology of Bacillus anthracis and its various manifestations in humans to the complexities of diagnosis, treatment, and prevention.
We've also explored the challenges posed by emerging forms of anthrax, like injection anthrax, and the potential threat of bioterrorism. And through the lens of that Hippo case study, we've delved into the intricate relationship between animal health, human health, and the environment.
And for you, dear listener, as a medical microbiologist, I hope this deep dive has not only reinforced your existing knowledge, but also sparked new questions and a desire to continue exploring the fascinating world of anthrax.
Because ultimately, the more we understand about this pathogen, the better equipped we'll be to protect ourselves, our communities, and the delicate balance of our ecosystems.
Well said. It's been a pleasure diving deep into the world of anthrax. Until next time, stay curious and keep those microscopes focused